from the very early stage, as a young scientist, she demonstrated outstanding qualities as a scientist. She published several papers as a first author. And then she moved to Worcester in the US, in the lab of Roger Davis, where she started working on JNK kinases in different tissues and the implication in metabolism and diabetes. And then again, she demonstrated her unique capacity to be an amazing scientist, publishing several papers, including a Science as a first author. I don't know when exactly, it was maybe 2009, it was when you established your own lab. When was that? 11. She moved to CNIO, where she became a junior PI. She got an ERC starting grant. And then since then, she's been building this amazing lab and amazing science, especially in metabolism with liver as a focus, but also expanding to other tissues and more recently into cardiac, into the heart. And as a last author, as a main author, she's published amazing papers, GCI, EMBOR, Nature, PNS, eLife. So an outstanding career, and that also explains why she has received many prizes, including the Prince of Girona, the EMBO Young Researcher, AstraZeneca, Banco Sabadell, European Society for Clinical Research. And as you probably know, she's very visible and she's been recognized one of the key scientists of Extremadura. She has recently received La Medalla de Extremadura from the Diputación of Badajoz. So we're very happy to have you here to host you. The stage is yours.
Thank you, Mariana. It's for me a pleasure to be here. It's amazing. The building, it was nice to be for first time here. I'm going to explain a little bit what we are doing in understanding how metabolism and how alteration of metabolism during obesity affect tumorigenesis development. Everything started when a long time back, when I was finishing my PhD, was described that in obese patients, stress kinases, mainly young one, was activated in the adipose tissue of these patients. It was thought that this activation was pathological, was driving the secondary effect of the disease. As I mentioned, this stress activating kinases belong to a big family, the stress kinases, that is the JNK family and the P38 family. And these kinases are important in the adaptation, help the cell to adapt to different stimuli. However, during obesity, where different triggers of this pathway are all the time present, like glucose, increased cytokine, increased free fatty acid in the blood, this pathway was all the time activated and became to be pathological. This was the beginning, and when I come back to Spain, we decide to also check whether the other leg of this pathway was also activated, and the answer was yes. The P38 pathway is also activated in the adipose tissue of obese patients. During my postdoctoral, we have found that this stress kinase, the activation of stress kinase in obesity, was very important for the crosstalk between adipose tissue and liver. And for that reason, we decide to focus in how obesity alteration, and how the alteration and the activation of this pathway during obesity will affect the liver diseases. And as I mentioned, we focus in how adipose tissue talk with the liver, how adipose tissue is alteration during obesity is going to affect liver homeostasis, but also how this activation of stress kinases appear during obesity in the liver, and how the activation in the inflammatory system, in the immune system of the liver is going to be important in the homeostasis of this liver. The first thing that we did was to try to understand the role of the less known pathway, the P38 gamma and P38 delta. The P38 pathway, when you hear about them, mainly everybody told about P38 alpha. However, almost nothing was done in P38 gamma and delta. This was, when I did my thesis, we were trying to understand the substrate of these kinases. However, when I start to try to understand what was the metabolism and the function of these kinases in metabolism, we didn't know so much. We found that these kinases are active in macrophages and control inflammation in the liver. But not only that, we also found that these kinases increase in the liver of patients with steatosis, and that appeared not only in obese, but also in lean individuals that have NASH in the liver. And we found an increase of the expression of this kinase. We decided to say what was this kinase doing during the development of this steatosis. This was the project of Tony in the lab, Antonia Tomás Loba. She arrived to the lab and she decided to understand how P38 gamma increased during the development of the different steps of the steatosis and the development of ACC. And she found that in humans, in the liver of human patients, P38 gamma, the expression correlates with the level of the fibrosis of these livers, suggesting that perhaps this kinase was important in the development of the disease and perhaps was involved in this process from the fatty liver disease, where the liver increased the level of lipids in the system, to the progression to NASH and to the development of ACC. That was the question she did. She asked herself whether P38 gamma was involved in this transition, and for in this paradigm to pass to NASH to hepatocellular carcinoma, you have to induce the proliferation in hepatocytes. You have to, hepatocytes are quiescent cells that they are very prone to be activated to induce proliferation if you induce damage. And that was the first question that she asked. Is P38 gamma important in the proliferation process of this hepatocyte when we induce a damage? And in mice, this question is more or less easy to answer because the liver, also in human, but the liver have this fascinating capacity of regenerate. If you cut part of one liver, the liver is going to, hepatocytes are going to sense that the whole organ is smaller and they are going to start to proliferate. And that was our question. If we cut part of the liver of the mice, P38 gamma get activated, is going to be important in this process, and that was the first answer. Yes, P38 gamma is present when we induce a proliferative damage in the liver. The second question was, if we take out P38 gamma in this hepatocyte, this is going to affect the proliferation of the liver, and the answer was yes. In BRDU staining, you can see how wild-type mice hepatocytes start to proliferate. However, mice that doesn't have P38 gamma in the hepatocyte cannot proliferate. They are stopped in quiescence. This signaling pathway was already known to be started by the phosphorylation of RB, and Tony checked how was the RB phosphorylation, and we see that nicely in a wild-type mice, RP is highly phosphorylated after the hepatectomy. However, when we don't have P38 gamma in the hepatocyte, this proliferation is blocked, suggesting that and the phosphorylation of RB is blocked. This phosphorylation, this RB, is a substrate of CDK. This paradigm of the serendipity is when I was doing my PhD, as I mentioned, we were looking for substrate of P38 gamma. And I always get complaint with this database because always that I find a new substrate, this substrate was described to be a CDK substrate. I was so unhappy with that. And I decided perhaps if you think about that, if every substrate that you find from a new kinase is predicted to be from another kinase, perhaps is that this active site is very similar. And that is what we try to demonstrate in this experiment. What we did is to find an inhibitor that binds to CDK1 and is not that bind to the active site. This means that this tells you not only that the inhibitor binds to this active site, is that this active site is similar. What we did with molecular computational biology is to check whether when we use the same modeling in the P38 gamma, we see the same. And that is what we see. What we see is that this inhibitor binds similar in CDK that in P38 gamma, suggesting that the docking site is similar and suggesting that these two kinases can phosphorylate the same substrate. Then we decide, okay, if that is, we can see this in much nicer way, how you can see that the inhibitor, you put it outside and you have to wait until go inside and find the place that is, because you don't force the inhibitor in the active site. And you can see that the Sylvia, the computational biology that did that, said you have to let the inhibitor to find the active site, and these two proteins get the inhibitor in the active site at the same time, suggesting that it's quite similar, these two active sites. Then the question was, is P38 gamma helping RB to the helping CDKs to the phosphorylation of RB? And that was the next question that we asked. And what we did is to get the full length of RB. We phosphorylate with the CDKs, and we also did the phosphorylation with P38 gamma. And what we found is that P38 gamma is able to phosphorylate almost in the same rate and in the same level RB in vitro. This, we did more experiments. We were able to demonstrate that P38 gamma can bind to RB and can phosphorylate also in vivo. If we put P38 gamma back in the knockout animal, we are able to phosphorylate RB. And what we hypothesize and what we think is happening is, and is quite normal if you think about that, is you need to induce proliferation in different moments in your life. Most of the proliferation that we have studied until now is induced by mitogen, because the cells need to proliferate. How when they already are proliferation, they need to proliferate. And these mitogens are going to activate the MAC cascade, the ERK, and this is going to induce the production of cyclin, and this is going to activate CDKs. But what happens if you have a proliferation induction, like in a damage, that doesn't have this cascade, because it's not, when you have a damage, when you have a NAS, you don't have a proliferative damage, you have a stress damage. Then you have to have another way of proliferate that is mitogen independent and is controlled by other pathways. And this is what we think that is happening in the hepatocyte, that we have two sets of induction, the classical mitogen cycling dependent proliferation, and also the cycling independent. And for that, what we have is a CDKs that need a cycling to be activated, and P38, both very similar, but this one that are activated by a stress signal. To really demonstrate that this is important for liver cancer, what we decided is to collaborate with people in the hospital of Salamanca, doctor of there, and we were able to demonstrate that P38 gamma is high expressed in human ACC. And when we try to evaluate whether the P38 gamma was important in the cancer development, what we see is if we don't have P38 gamma in hepatocyte, if we induce tumor in these mice, we have less number of tumor, less tumor size, and the mice are going to live more because they don't develop tumor, suggesting that P38 gamma is necessary for this initial of the proliferation stage in the tumor, and that inhibiting P38 gamma is going to be a new target for the treatment of liver cancer. Summary, what we see is there are alternative pathway that is a stress pathway independent of mitogen that is going to activate P38 gamma, that is going to phosphorylate RB, and is going to induce the initiation of the cell cycle in the hepatocyte, and is going to induce the tumor. We think that because this pathway is activated by stress, is more independent of mitogen, is going to be less, is going to have less secondary effect, because this pathway, and we demonstrate, is not, for example, important in tissues that there are already proliferative, like the intestine or other tissues, because in this proliferation of these tissues that have to proliferate, the proliferation is not driven by a stress stimulus, it's a mitogen proliferation. We next start to look for the rest of the liver tissue and for the inflammatory profile and immune system that is inside of the liver. And we started by this, the neutrophils, perhaps because Andres Hidalgo was in that time in the CNIC, and I hear all his talk and became interested in what's happening with neutrophils. And thanks to our collaboration with Andres and the work of Barbara, Nuria, and Ivana, we were able to demonstrate that P38 is very important for that, for the movement, for the infiltration of the neutrophil in the liver. Neutrophil is the first immune cell that are going to go to the liver and are going to induce the NAS, the steatosis. It's going to change the metabolism of this liver, and we demonstrate that when neutrophil gets infiltrated, they are going to change the metabolism of the hepatocyte, and it's going to induce lipogenesis in this liver with increased lipid droplet. And we find that because we didn't have P38 gamma and delta in the neutrophils, these neutrophils were not able to migrate. They don't arrive to the liver, and for that reason, these mice were protected against NAS and non-alcoholic fatty liver disease. In that time also was starting to be told that neutrophils infiltration in the different tissues were very important for homeostasis, and that this neutrophil infiltration in the tissues have a circadian rhythmicity. We have this capacity to to study that because we knew that P38 gamma and delta were important in this infiltration. And the next question that all of them did it was very long, was whether this infiltration of neutrophil in a circadian way was important controlling liver metabolism and circadian regulation of liver metabolism. The first thing that we did is we used our mice, the mice that cannot have high infiltration of neutrophil when we damage the liver, for example, with MCD diet that induces steatosis and NAS, or with high-fat diet. We did with both, and we found that the clock gene are altered. The BMAL is down, and the opposite one are up. The PER, TRI, and REP are up, suggesting that the lack of neutrophils in this liver is affecting the clock of these livers. And the next question is this neutrophil infiltration in the liver is circadian? And whether this circadian infiltration of neutrophil is important to maintain the clock gene in this liver. And that what the first thing that we did, we evaluate the neutrophils that were infiltrated in different times of the day and the night. You can imagine the one that have to go at 2 o'clock in the morning. That is the reason we have several ones. This day and night change was correlating with a change in the clock gene BMAL. And that not only correlates with the clock gene, but also with the metabolism of this liver, suggesting that neutrophil infiltrating the liver in a circadian way, this control the clock genes in the liver, and this is in some way affecting the metabolism. Because at the end, the regulation of the clock is important to the liver be adapted to the change that have during the day. The liver have to know before we eat that we are going to eat, to be prepared. That at night, the liver need to know that he is going to be starving for a long time, to be prepared, to be accumulate before that happen enough energy. That means these three things have to be tightly regulated and have to be regulated in a way. And that's what we wanted to understand. For try to understand that, we decided to use neutropenic mice that doesn't have neutrophils, and check whether if we don't have neutrophil, these changes that appear, for example, with the MCD, they go back. And what we see was that when we dysregulate the liver with MCD, with dysregulate, BMAL go up, the clock, and that is blocked when we don't have neutrophils, suggesting that neutrophils is implicated in this process. And this was also affecting the metabolism and the capacity of the liver to be steatotic. We have more lipid droplets when we have neutrophils than when we don't have neutrophils. The next question was, how is mechanistically dysregulated? How neutrophils told to hepatocytes what they tell them to change these clock genes. We were so surprised that when we incubate neutrophils with hepatocytes, we see a change in BMAL as what we expected, because BMAL goes down when we don't have neutrophil. When we have neutrophil in vitro, we have upregulation of BMAL. And after checking a lot of signaling pathways, I was happy, and I'm happy to go again to the JAMP pathway that I have for a long time forgotten in my new lab. And we found that these neutrophils increase the phosphorylation of JAMP in hepatocytes. We decided to check what was secreting neutrophil. We checked for different factors that neutrophils can secrete. And finally, we found that elastase was doing the same effect. When we put elastase, that is a protein that is secreted by neutrophil, we see the same effect. We see an increase of clock and activation of JAK, suggesting that what is happening is neutrophils get infiltrated in the liver in a circadian way. These neutrophils are going to produce elastase, they are going to induce the JAK activation, and they are going to increase PMO. We did this also with the elastase knockout mice to demonstrate that was this the pathway. And we also did this with the JAK knockout mice to demonstrate that when we don't have JAK, we cannot do this pathway. In a summary, what we found is that neutrophils are going to be infiltrated in the circadian way in the liver. Once they get infiltrated, they are going to produce elastase, and they are going to activate TNK. And that, we also demonstrate that, is going to inhibit the production of FGF21 in the liver. FGF21 is a very important hepatokine that is going to control also full body homeostasis. And this is going to change the metabolism of the liver and the clock genes of the liver. When we are young, there are less neutrophils infiltrated in the liver. JAK is going to be less phosphorylated. Then it's going to be more FGF21 and less PMAR and triglycerides in this liver, suggesting that this is a homeostatic process to be able to control the liver metabolism. And if this is a homeostatic process, and if we don't have JAK, we don't have this process. That was the next question. And the answer was yes. When we don't have JAK, this regulation of clock gene is completely destroyed. Because of that, we decide to try to understand whether this dysregulation of the circadian rhythm in hepatocytes, because at the end, this is telling us that these hepatocytes are not able to know when they are day, when it's that night, when they have to do lipid accumulation, is going to affect liver cancer development, because have been linked that if you are have a problem in your circadian system, you are going to develop liver cancer. And that was the early questions he asked, whether these mice that have altered this circadian clock, they have they were more prone to develop liver cancer. And the answer was, first of all, when they get older, without doing anything, they have more blood bile acids. They have alteration in the bile acids production. They produce more toxic bile composition, and they develop cholangiocarcinoma. This is like, we don't do anything in these mice. They do this by their own. And we found that this cholangiocarcinoma is induced by the dysregulation of the very similar hepatokine that is FGF15. And we are now trying to understand how is this dysregulation, and whether FGF15 and FGF21, even we think could be beneficial to protect mice against steatosis, could have deleterious effect if we give them in long term. Because remember, if you don't have TNK, you are going to have a lot of FGF21. And what we found here is this could affect the proliferation, because these FGF hepatokines are going to induce the proliferation of cholangiocytes. And I'm going to summarize what we know now of the liver hepatocyte. What we know is that if we deplete P38 gamma and P38 beta, this was known. We have activation of CHAN1 and CHAN2, and that is going to affect the HCC. If we deplete GNK1 and GNK2, we are going to induce cholangiocarcinoma. However, if we deplete P38 gamma, we are going to protect against HCC, suggesting that in the three, because these three pathways have been, a lot of companies have been trying to do inhibitors to try to prevent ACC. But we really think that the only one that we have to, could be a good target is P38 gamma. And we think that we have to be very careful in the way of inhibiting this P38 gamma to avoid to affect the other leg of the pathway. And we will try to do this now in the CNIO. The other cells that we can find, and they are very abundant in the liver, are the Kupffer cells, monocyte and Kupffer cells. Once we have a liver that is going to develop steatosis, these Kupffer cells are going to start to die, and monocytes are going to get infiltrated in the parenchyma of the liver, and they are going to become similar to Kupffer cells. It's an active process that we are trying to understand now, how these monocytes that came from the blood get transformed in monocyte-derived Kupffer cell, and they are important in the NAS, but also how embryonic Kupffer cells are important for this process. And the first thing that we decided to do is to try to inhibit the full pathway, because at that time we knew that P38 alpha and beta, if we block, is going to affect JAM and P38 gamma and delta. And we want to really block in inflammatory cells the full pathway. And there was another reason, is that if we get an inhibitor of this pathway, we are going to have the kinesis in the system. What we are not going to have is the activity. For that reason, we decide to have the kinesis in the system, but to block the upstream kinesis, the NTP3 and the NTP6. And we did conditional animals that they don't have NTP3 and NTP6 in the myeloid compartment. We found that they don't have in the bone marrow, but also Kupffer cell doesn't express NTP3 and NTP6. That means we don't have in the full bone marrow. And for us, we were expecting that if we don't have any longer stress activation in these Kupffer cells, we are going to protect the mice against the deleterious effects of obesity. We were not expecting to have changes in body weight, but the less that we were expecting was to have the opposite change, that the mice are more obese and they have more body weight and more fat mass. How immune cell and macrophage can affect this in the animals. We put the animal in metabolic case, and we found that these animals have lower energy expenditure. If we check in the metabolic case, this means that this animal, for being like that, are spending less energy. That means they have more energy to get obese. And what we found is this was not due to they do more exercise or less exercise. What they do is they have less body temperature. In the brown adipose tissue, you can see here in white, more white means more temperature, that the wild type have much more temperature than the knockout, suggesting that this lack of stress activation in the myeloid compartment was affecting the brown adipose tissue temperature. And in that time, Frances Villarroya here was described that macrophage infiltrating in the brown was important for doing this effect. We were trying to understand whether these macrophages infiltrated in the brown adipose tissue were different, but we didn't find anything. We were a little lost how you can affect ground temperature if you don't change the infiltrated macrophages here. And finally, what we were doing, because we phenotyped mostly all the mice to see what was all the effect, is where we were quite surprised is the huge steatotic liver that these mice develop. They have a huge lipogenesis in this liver. Most surprised at that is that FGF21 was very, very low in this liver. And not only that, if we look for the protein, was very down, and in the blood was also very down. Was described that FGF21 is a hepatokine that can control thermogenesis in brown adipose tissue, and that was our next question. Is P38 activation in the macrophage affect the production of FGF21 in the liver, and this could be affecting the steatosis because lack of FGF21 is going to induce acute steatosis, and whether this is going to also affect the thermogenesis of these mice. And to answer this question, what we decide to do is to use mice that doesn't have FGF21 in the liver, only in hepatocyte, and the mice that and take the bone marrow from mice wild type and from mice that cannot activate P38. We put this bone marrow in these two types of mice, and then we have 4 groups. Mice that have FGF21 in hepatocyte, but have or don't have P38 activation in the immune system, or mice that doesn't have FGF21 in the hepatocyte and have P38 activation or not P38. No? What we see is follow when you have FGF21 in the liver, in the hepatocyte, you see the same. You don't have P38 activation in macrophage, then your body weight go up, your energy expenditure go up. But if you don't have FGF21 in the hepatocyte, if you cannot respond, then we don't see any difference in body weight or energy expenditure. And the same happens with the vacuum therapy. What we see here is if you don't have P38 activation in the myo compartment, what you see is temperature goes down to the level of a FGF21 knockout in hepatocyte. This is similar. However, if you don't have that in the system, if you don't have FGF21, lack of P38 alpha doesn't do anything more. We were trying to, again, understand how these two cells communicate, what macrophage signal to hepatocyte to really induce this chain in FGF21. And what we did is to extract embryonic Kappa cell in vivo from the control mice and from the mice that cannot activate P38. And the third cytokine that was here was interleukin-12, and was at that time described as interleukin-12 is a very important signal for hepatocyte. And we decided to check this. Doing a long story short, what we found is that P38 inhibits the production of interleukin-12. That means when we have a macrophage that cannot activate P38, we are going to have huge amount of interleukin-12 that is going to inhibit FGF21. FGF21 is very important for the hepatocyte adaptation. It's going to block the steatosis, but not only that, it's going to go through the blood to the brown adipose tissue, inducing thermogenesis. Because we don't have P38 activation in macrophage, at the end, what we have is huge steatotic liver and increase of body weight. The last thing that I'm going to explain is the other part of this mess in immune cell is T cell, because when we started long time back, but I am not an expert in T cell. Ivana arrived to the lab and she was working in T cells. And we decided to understand what P38 was doing in these T cells. And in that moment, it was known that T cells were important in the adaptation of adipose tissue to obesity, and that in lean individuals was a regulation of T regulatory cells that disappeared when we became obese. The first thing that Ivana do with the hair of Rafa, that was a PhD student that became a bioinformatic now, was to check in database of single cell analysis of human adipose tissue that there are alteration in T cell of the expression of the pathway, the P38, the phosphatase, and the kinases. ATP6, P38 alpha, ATF2 is the downstream substrate. We have a downregulation of the phosphatase and upregulation of the kinase, suggesting that what we have is a activation of this pathway in T cells in human, in vivo, during obesity. Then what we did is the same strategy. We block all the pathway with a double knockout, but now in CD4, using CD4-3 animals. Remember that this animal is going to deplete our pathway, not only in CD4, but also in CD8, because CD8 came before from cells that produce CD4. That means we don't have NTP3 and NTP6 in CD4 and in CD8, but we have it, for example, in AKT and other types of tissue. We decide to put these mice in high-fat diet, similar to what we did before. And what we found is these animals are protected against obesity. They have less body weight. And as you can see here, the amount of fat that they accumulate is much more lower, suggesting that the lack of P38 activation in T cells protect mice against obesity. We check also if they are protected against diabetes and they have less glucose in fed condition and in fasted conditions, suggesting that they have less hyperglycemia. We again check for the temperature to check whether these changes in body weight could be induced by changes in the metabolism of the adipose tissue. And what we see is that lack of P38 activation in T cell increase the bath temperature, that you can see here in the imaging how these mice are obese. This is more linear, this has more temperature, this has less. If you check in the histology, the brown adipose tissue, you can see how this is a more active brown adipose tissue with a lot of cellularity, less fat accumulation. They have more UCP1 expression, that is the inductor of thermogenesis, and the genes that are known to be involved in the thermogenic pathway. That means we now know that if we don't have P38 activation in T cell, what we have is a thermogen pathway in the brown adipose tissue. And we also check that this happens in the white adipose tissue, that this white adipose tissue, you cannot measure the temperature, but we can measure the genes that they produce. They are more beige adipose tissue. And then we start to work in what happened in this adipose tissue, what happened in the white and in the brown adipose tissue. And we found that this adipose tissue have higher number of T regulatory cells in white and in brown tissue. This is white, because in white you have more cells, more easy to measure. And this T regulatory cell, what we see is that they produce this is the pathway of interleukin 35, that is a cytokine that is have different genes produce this cytokine. And what we see is these T regulatory cells are going to be more, and they are going to have more production of interleukin 35. That is a good cytokine for the during the leaves. We decide to check in human cell if that happens, and whether if we inhibit P38, we have more interleukin 35 in the cell, and what we see is this: we have more production of interleukin 35 if we inhibit P38. We try to understand what is P38 signaling in this cell, and what we found is that when we don't have P38 activation, and you can see here how nice we can block P38 activation when we don't have NKG36, we have a huge activation of mTOR pathway. We decide to check whether this activation of mTOR pathway was implicated in the pathway with the cell of Alejo that was nearby in the CNIO at the time. And what we found that is that if we put rapamycin in this cell, we have an inhibition of interleukin 35 in this T regulatory cells, suggesting that P38 activation block this mTOR pathway, and for that reason block interleukin 35 production, suggesting that if we block P38, we can have more interleukin 35. The next question, and perhaps this was the question more difficult for us to answer, was: is interleukin 35 the driving force of the browning? For doing that, what we decide is to use recombinant interleukin 35 and put in wild-type mice, and we found that after 4 hours, we have a higher bad thermogenesis in this animal. We really need to, we are now generating specific nanoparticles to really try to increase the level of interleukin 35 in a more chronic way, because here we only know that in a short period this is working, but we need to really know if this is working in a long state. As a summary, I put it here. What we have is that during obesity, P38 in T cell get activated, and the activation of P38 in T regulatory cell is going to reduce the number of T regulatory, is going to the number of T regulatory cells that are in the adipose tissue, but also the function of this T regulatory producing interleukin 35. And when we don't have P38 activation, then we are going to have more interleukin 35 in the mice. This is going to activate the browning of these adipocytes, and is going to increase the thermogenesis and protective pathways that are going to be beneficial for the this animal, to protect them against obesity. And now what we are doing now is to try to understand whether this is important for the liver cancer development, using these mice to induce liver cancer, and to try to understand whether these T cells are more prone to be exhausted before of time, because they are more active. And with that, I want to thank all the people of my lab, the people that helped us before in the CNIC. Was a very nice time there. I'm very happy to be now in the CNIO, to be able to collaborate and start a new life in a new institute. Thank you very much.